Student, in this lab, I'll explain the antimicrobial sensitivity test. This is an important laboratory test and done after identification the species that are responsible for infection. Antibiotic sensitivity test is a laboratory test that measures the ability of the antibiotic to kill or inhibit the growth of the bacteria. Antibiotic is a chemical produced by microorganisms like bacteria and fungi that are designed to kill or inhibit bacteria growth. You should understand the difference between antimicrobial and antibiotic. Antimicrobial is a broad term, includes antibiotic and other agents that act against bacteria, fungi, and viruses, while the antibiotic is a chemical agent designed to kill or inhibit bacteria growth. But when we use this antibiotic in a wrong way or too much use this antibiotic can lead to the antibiotic resistance. It means the bacteria survive and the drug becomes less affected. Therefore, the antimicrobial sensitivity test is essential to choose the most correct antibiotic and maintain the effective treatment. And before I explain the method of this test, there are some important terms such as bactericidal, that means the antibiotic directly kill bacteria, for example penicillin, vancomycin, whereas the bacteriostatic is the agent they don't kill bacteria but just inhibit their growth. It means they stop the growth of the bacteria such as the tetracycline. And the inhibitory concentration, it means the smallest amount of the antibiotic needed to stop bacteria growth, while the minimum bactericidal concentration is the smallest amount needed to kill bacteria. So this difference between two terms is important to understand. Minimum inhibitory concentration is the smallest amount of the antibiotic that inhibits growth of the microorganism, while minimum bactericidal concentration is the smallest amount that kills the microorganism. The method of the antimicrobial sensitivity test, the most common one, is diffusion method, also called Kirby power method, that measures the bacteria growth around antibiotic test. Other methods include quantitative e-test that determine the concentration of the antibiotic needed to stop the growth of the bacteria, and dilution test, which use different antibiotic concentration to find the minimum inhibitory concentration. In this lab, I will discuss the diffusion method and the quantitative e-test. Now, the most common laboratory technique, that is diffusion method, that are also called Kirby power method. And in this test, we use a special media that are called Muller-Hinton agar and measure the sensitivity of the microorganism to antibiotic. The principle of this test, we put the antibiotic disc on a agar plate that has been inoculated with bacteria and put the plate in the incubator. After that, if bacteria are sensitive to antibiotic, a clear area that are called zone of the inhibition will appear around a disc where no bacteria can grow. And this zone of the inhibition, it measures across a diameter. And why we use the Muller-Hinton agar for this test? The first point, because it's non-selective and non-differential media. Non-selective for a specific type and cannot differentiate between bacteria group, it allowing all bacteria to grow on it. We use this media because all bacteria can grow on this type of media. The second point, it contains starch that absorb the toxin may released by bacteria and interfere with antibiotic. The third point is a loose agar structure, because this structure, it allows for better antibiotic diffusion and better inhibition zone will appear on this type of media. That's why we use this media for sensitivity. And the procedure of this test, first we make the suspension of the bacteria by transfer the colony from the pure culture in a sterile cotton water. and this suspension should have the concentration 0.5 according to the McFarland standard. And the media used in this test, Muller-Hinton agar, we should have the Muller-Hinton agar, which is a special. Prepare the suspension of the bacteria and Muller-Hinton agar. We dip the sterile swab into the bacterial suspension and make the streaking over the entire surface of the Muller-Hinton plate. Using a sterile forceps, we put the antibiotic disc on a agar plate and pressing them gently. Let agar to be dry 3 to 5 minutes, then incubate it at 35 to 37 Celsius for 60 to 80 hours and put the plate in the incubator in the inverted position. Now the procedure of the antibiotic sensitivity test. We do this test after identify the correct identification of the microorganism, the species of the bacteria. We should have the pure colony of bacteria, fresh culture. We have the special media, which is called Muller-Hinton agar, special for this test. Antibiotic disc according to the type of the bacteria, sterile swab, sterile loop, and sterile forceps. First, we make the bacterial suspension. We need the McFarland standard 0.5. Or measure the turbidity in the turbitometer. If we don't have the turbitometer, we make the suspension and compare with McFarland standard. We put 2 to 3 mm of normal saline, and by sterile loop, we put 1 to 2 colony of bacteria mixed with the normal saline, 2 to 3 mm of normal saline. As I mentioned, if we have the turbitometer, we measure the turbidity, and the turbidity should be 0.5. If we don't have the turbitometer, we compare with a McFarland standard, which is special standard 0.5. If we compare both of them similar to each other, the turbidity of both. After prepare 0.5 suspension of the bacteria by sterile swab, we put on the suspension of the bacteria and make the spreading on the Muller-Hinton agar plate. Spread the bacteria evenly over all the agar plate. And all the work is near the burner because the sterilization is very important. After spreading the bacteria evenly over all the agar plate, using sterile forceps, sterilize by alcohol, then burner, allowing to cool, then put the antibiotic disc, and we should put the antibiotic placed evenly at least 25 mm apart and 50 mm from the plate edge to prevent overlapping zone and accurate measurement of the inhibition diameter. After putting, pressing gently and allowing to be dry 3 to 5 minutes. Then put into the incubator at 35 Celsius for 60 to 80 hours in a inverted position. After we measure the zone around each antibiotic disc, and I will go to show you how to measure the diameter of the zone of the inhibition. In the next slide, I explain all information about measurement the zone. And after incubation, antibiotic diffuse into the agar, and if the organisms are susceptible, a clear zone of inhibition appears around a disk. If resistant, no clear zone is formed. Following incubation, measure the diameter of each inhibition zone in millimeters using the ruler placed on the upside of the closed plate. And we should measure the entire diameter of a clear zone, including the disk. For example, image you can see on a disk we show the large clear zone which indicate the susceptibility, whereas another disk show no clear zone which indicate the resistance of the microorganism. And the major inhibition zone diameter compared with a standard breakpoint provided in CLSI table, Clinical and Laboratory Standard Institute, and according to this criteria, results report as sensitive, intermediate, or resistant that help to select a correct antibiotic for treatment. For example, using ampicillin for gram-negative bacteria and enterococci, according to this criteria, the sensitivity reported when the zone of the inhibition is larger or equal to 70 mm. But if the zone of the inhibition is smaller or equal to 60 mm, then the result reported as a resistant. And when we select any antibiotic, it's essential to consider the bacterial type, the site of the infection, and we should know the susceptibility profile. For example, if Escherichia coli from the UTI infection, urinary tract infection, show the resistant to ampicillin but sensitive to ciprofloxacin, then the ciprofloxacin we should choose as. And as I mentioned, the result reported as sensitive, it means the antibiotic inhibit bacteria growth at a standard dose. And intermediate, it means bacteria show the reduced susceptibility, the higher dose may be needed. And resistant, it means the antibiotic does not inhibit bacteria. It means the microorganism is resistant to this type of the antibiotic. Another method of the sensitivity test is E-test or absolometer test. It's a quantitative method used to determine the minimum inhibitory concentration of any antibiotic. A strip containing gradient antibiotic concentration that put on an inoculated agar plate, allowing the drug to diffuse and form elliptical zone of inhibition after incubation. The minimum inhibitory concentration is read at the point where the edge of the inhibition ellipse intersects the scale on the strip. For example, if the intersection happens at 0.25 microgram per milliliter, then the minimum inhibitory concentration is 0.25 microgram per milliliter, as you can see here in this image.